Welcome to the deep dive. Today we're diving into, well, a really fundamental challenge in medicine.
Mm.
How do we actually figure out if a new drug is going to work?
Mm.
And, importantly, who it's going to work for?
Yeah, it's a huge question. And historically, the approaches have been a bit limited, haven't they?
Definitely.
Uh,
Often just looking at one type of biological data, right?
yeah,
Like maybe just the genes or maybe just focusing on proteins.
exactly. And you can imagine how that gives you, well, an incomplete picture.
But trying to see a whole painting by just looking at the blue bits.
Precisely. You miss the context, the interactions.
Which is why there's all this buzz now around multi-omics data.
Right. Looking at everything together.
Yeah, the whole biological landscape. So
Yeah.
we're talking about the genome.
Mm.
That's your DNA blueprint, basically.
Okay.
Then the transcriptome, which genes are actually switched on, making RNA.
Right. The activity level.
Then the proteome, so all the proteins being made, being modified,
Yeah.
often the actual drug targets.
Crucial layer, that one.
And finally, the metabolome. All those small molecules involved in, metabolism, how the drug gets processed.
Each layer gives you a really unique perspective. Like, mutations in the genome can point to what's driving the disease.
Okay.
While, say, the transcriptome shows if a drug is affecting the target gene's activity.
And the proteins show the target's actual state.
Exactly. And metabolites can tell you if the drug is even being broken down correctly by the body.
It's a lot of different information sources.
It is. And what's really fascinating now is some new research using all of this. There's a deep learning model called MAG-NET.
MAG-NET, okay. Multimodal adaptive graph neural network, is that it?
That's the one. And the whole idea is to take all this rich multi-omics info and use it to get much better predictions about drug efficacy.
Especially for specific tumors you mentioned.
Yes, specifically trying to predict how well a drug might work against certain cancers,
Right.
moving beyond that single snapshot view.
Right, towards a much more complete picture. Okay, so predicting drug efficacy is already super complex. But you mentioned a roadblock earlier. The data.
Ah, yes. The labeled data problem. It's a major bottleneck.
Meaning we often just don't have much information saying this drug worked or this drug didn't work for new experimental treatments.
Precisely. You need that labeled data, the known outcomes, to train the AI models effectively. But getting it means, big, expensive clinical trials.
Which isn't always feasible, especially for rare diseases or specific patient groups.
Exactly. So you end up with this scarcity of really good labeled data to learn from.
So how does MAG-NET start to get around that, this lack of known results?
Okay, so this is where it gets clever. It uses a technique called self-supervised learning.
Self-supervised learning. Can you break that down a bit?
Sure. Essentially, it lets the model learn useful patterns from the input data itself without needing tons of those work didn't work labels initially.
How does that work?
Think of it like giving the model parts of the data and asking it to predict other parts. It generates its own sort of temporary labels, pseudo-labels, based on the internal structure and relationships within the multi-omics data.
Ah, okay, so it's like learning the rules of the game before you know the final score.
That's a good analogy. It trains on these self-generated tasks first, learning the underlying biological patterns. Then later, you fine-tune it with the real but limited labeled data you actually have.
And that initial self-learning helps it generalize better, make predictions on new things it hasn't seen.
Exactly. It builds a much more robust understanding of the biology first, which makes it better prepared to use the scarce labeled data effectively.
That is a smart way to tackle the data issue. So we know MAG-NET uses these different omics layers. How does it actually pull them together? It can't just be mixing them all up randomly, right?
No, definitely not. It's very structured. Each type of omics, data genomics, transcriptomics, proteomics, metabolomics gets processed individually first. So take genomic data mutations, copy number changes, things that drive disease. The model turns this information for each sample into a numerical format. And embedding.
Like a digital fingerprint.
Kind of, yeah. A compressed representation that captures the key features. For MAG-NET, it's a 512-dimensional embedding for each omics type.
And it does that for the gene activity, the protein data, the metabolite data too?
Yes, exactly the same process. Each gets its own 512-dimensional fingerprint.
And then?
Then, these individual fingerprints are combined. They're concatenated, basically joined end to end, to create one big integrated feature vector, 2048 dimensions in total.
So that vector represents the combined snapshot from all the omics layers for that sample.
Precisely. It's like merging those individual photos into a detailed panorama.
Got it. But the name, graph neural network, Where do graphs come into play?
Right, this is the other really cool part. It doesn't just use those vectors. MAG-NET also represents each omics dataset as a graph.
A network.
Exactly. Imagine nodes in the network are the biological bits like genes, proteins, metabolites.
Okay.
And the edges, the lines connecting them, represent relationships. So in the genome graph, an edge might mean two genes often mutate together.
I see. And for proteins?
In the proteome graph, edges could show known protein interactions, or maybe which protein a drug targets. Same idea for metabolites, how they link up in pathways.
So it's capturing the connections within each biological layer, not just the levels, but the interactions,
That's the key insight. Biology is a network. Things interact. You need to model that.
which is where the graph neural networks, the GNNs, are useful because they work in networks.
Precisely. GNNs are specialized AI tools designed exactly for this kind of interconnected, graph-structured data. They're really good at learning from these relationships.
And MAG-NET uses specific kinds. You mentioned GCNs.
Right. Graph convolutional networks, or GCNs. The basic idea is that a GCN updates the representation of a node, say a gene, by looking not just at the gene itself, but also at its immediate neighbors in the graph.
So understanding a gene requires looking at its close connections. Makes sense.
It does. It aggregates information from the local neighborhood.
And the other type was gates, graph attention networks.
Yes, gates add another layer of sophistication. They learn to assign different levels of importance, attention weights, to different neighbors. Meaning the model figures out that for understanding this specific gene's role, its connection to neighbor A is maybe way more important than its connection to neighbor B.
So it focuses on the most relevant connections within the network.
Exactly. It leads to more refined and accurate representations or embeddings for each node.
And MAG-NET does this GCN and gate process for each omics graph separately, genome, transcriptome, etc.
That's right. It learns the unique network patterns within each data type independently first.
Okay, this is getting intricate. So you've processed each omics layer, turned it into embeddings, built graphs, analyzed the connections with GNNs. Now, how do you bring the insights from these different omic perspectives together?
Right, the integration step. This is another place where MAG-NET is quite innovative. It uses another attention mechanism.
Attention again.
Yes, but this time it's working across the different omics types. After the GNNs have processed each graph, the model learns to weigh the importance of the resulting embedding from each omics layer.
So it's deciding how much to listen to the genome data versus the proteome data, for example.
Exactly. It might learn that for predicting response to drug X, the proteomic network information is the most critical signal. Or maybe for drug Y, the metabolic pathways are key. It assigns weights based on relevance for this specific prediction task.
And then combines these weighted insights.
Yes, these weighted omics embeddings are then concatenated, joined together into that final comprehensive representation that captures the most salient information from across all the biological layers.
So it's adaptively focusing on the most important data types for the specific drug and context.
That's the adaptive part of the name, essentially.
Okay, and this final representation is what's used to predict if the drug works.
Correct. This integrated embedding is fed into a standard component in AI, a fully connected layer, which makes the final prediction.
And that's a yes-no prediction,
Yeah.
responsive or non-responsive.
Basically, yes. It's framed as a binary classification problem. To train it, they use a common loss function called binary cross-entropy that just measures how far off the predictions are.
Okay.
And the very final output uses a sigmoid function, which squashes the result into a probability between 0 and 1. Like a 0.9 score means a 90% predicted probability the drug will be effective.
Wow. Okay. That's a complex pipeline.
Hmm.
But theory is one thing. Did they actually test this with real data?
Oh, yes, absolutely. They used a whole range of well-established public data sets.
Yeah.
Things like TCGA and COSMIC for genomics,
I'd heard of those.
GEO for transcriptomics, CPTAC and proteome exchange for proteomics, and the metabolomics workbench for metabolite data. So real complex biological data.
And they focused on specific cancers and drugs.
Yeah, they looked at examples like lung cancer and breast cancer and tested predictions for common chemotherapy drugs like cisplatin and paclitaxel.
So the big question, how did MAG-NET actually do compared to other methods?
The results looked really promising, actually. They compared it not just to models using only a single omics type, but also to another multi-omics model called Deep Drug.
And
MAG-NET consistently performed better. Its accuracy reached 86.3%.
okay. How good is that?
Well, that was a 6.1% improvement over the existing deep drug model, which is pretty significant in this field.
A 6% jump is quite a leap.
It is. And they also looked at another metric, AUC area under the curve, which is a good measure of overall predictive power. MAG-Net got 0.93.
Which was better than deep drug.
Yes, 0.06 higher. So across different metrics, the data suggests MAG-Net provides superior prediction performance.
That difference in accuracy, even if it sounds small numerically, could mean a lot in practice, couldn't it?
Absolutely. In drug development, improving prediction accuracy like that could save huge amounts of time and resources by focusing on more likely candidates and ultimately help patients faster.
Okay, the results sound good. But, one common critique of these complex AI models, especially in biology, is the black box problem. We get an answer, but we don't know why.
A very valid concern. And the researchers tackled this head on. MAG-Net was designed with interpretability in mind.
How so?
Well, remember that attention mechanism that weighs the different omics types?
Yeah, deciding which data layer is most important.
You can actually visualize those attention weights, create heat maps showing for a given prediction how much focus the model put on genomics versus proteomics versus metabolomics, and even which specific features within those layers were most influential.
Ah, so you can see what it's paying attention to.
Exactly. And furthermore, you can analyze the embeddings that the GNNs learned for the individual node, the genes, proteins, metabolites.
What does that tell you?
By visualizing these embeddings, maybe using techniques like TSNE, you can see which biological components cluster together in ways that relate to drug response. It can help pinpoint specific genes or pathways the model thinks are critical.
So it's not just saying yes or no. It's potentially highlighting the biological reasons behind the prediction.
That's the goal, to provide clues about the underlying mechanisms, which is incredibly valuable for understanding the drug, the disease, and why some patients respond differently.
That moves it beyond just prediction into potentially generating biological hypotheses.
Precisely, which opens up some really exciting potential applications.
Like what? What are the big hopes for a tool like MAG-Net?
Well, the obvious one is in new drug development itself, helping pharma companies prioritize candidates, maybe increasing the success rate of clinical trials.
Okay, that's huge, financially and ethically.
Definitely. Then there's personalized medicine. Imagine using a patient's specific multi-omics profile to predict which available drug is most likely to work for them.
Truly tailoring treatment.
Yes. And also, in basic research, using it to discover new biomarkers, indicators of drug response, or just getting deeper insights into disease mechanisms by seeing what the model flags is important.
It sounds like it could touch many points in the medical pipeline.
It really could. It's all about making the process more precise, more personalized, more efficient.
So what's next for this line of research? Where does it go from here?
The paper points to several directions. One big one is more validation, testing it on more cancer types, more drugs, to really prove how generalizable it is.
Makes sense. Broader testing.
Right. Another idea is integrating even more external biological knowledge, like plugging in known pathway databases or protein interaction networks directly into the model structure.
Leveraging existing knowledge.
Exactly. They also mentioned some pretty advanced ideas, like using reinforcement learning to figure out optimal combinations of drugs.
Wow, okay. That's complex.
It is. And other things like maybe incorporating time-lapse data, how things change over the course of treatment, or validating predictions against real clinical trial data, and just generally keeping up with improvements in AI itself.
Constant refinement.
Always. It's a fast-moving field.
Well, this has been a really illuminating deep dive. It's genuinely fascinating to see how these incredibly complex AI methods are being applied to such a core problem in medicine.
It really shows the power of combining computational approaches with deep biological understanding. MAG-Net and tools like it feel like a significant step forward.
Using AI to untangle the sheer complexity of biology and drug interactions.
Yeah, by intelligently bringing together all these different layers of information, the genome, the transcriptome, proteome, metabolome, it offers a much richer, hopefully more accurate way to predict how therapies might work.
The potential impact on speeding up drug development, making it more personalized, is pretty profound.
It really is. Ultimately, the goal is better outcomes for patients.
Absolutely.
Right.
And it leaves you thinking, doesn't it? As these predictive tools get even better, how fundamentally could that shift the entire landscape? Not just pharmaceutical research, but how we approach treating individuals with diseases like cancer in the coming years. It raises some big questions to ponder. Thanks for joining us for this deep dive.